Good morning, everyone. Thank you for coming. Thank you for the invitation. It's very fun to be here. So as David said, I'm not going to be showing you how to design small molecules. I'm more talking about the biological context where these targets come from in the first place. So just by way of background, what my lab does, we do a lot of machine learning models to understand the regulation of gene expression in mammalian cells. And so this is sometimes called regulatory genomics, or the genomics of gene regulation. So that's, can you see this, my cursor? Maybe not. Okay. But we also work with collaborators in preclinical models to study basic immunology, autoimmunity, and cancer immunology. And we also work with collaborators in cancer epigenetics. So these are cases where you have somatic alterations in epigenetic regulators that change the whole state of cancer cells, and you need additional tools to understand what's going on. Okay? But today I'm going to talk about sort of machine learning models for regulatory genomics. All right, so I'm going to start with explaining just the basics of gene regulation and then motivating why you should care about gene regulation if you are, trying to design drugs and use AI, okay? So I don't know what people's background is, but hopefully you have a picture in your mind like this of what's the information in the nucleus of the cell, right? So you probably know that genes, the gene bodies, the protein coding part of the genome, gene bodies are about 10% of the genome. The actual coding part of genes is about 2% of the genome. And then we have a lot of non-coding DNA. And there are specific elements, regulatory elements, where proteins called transcription factors bind. Some are near the gene, so these promoter regions. And some might be very far in the linear genome, a megabase away perhaps, and they're called enhancers. But they're looping in 3D, making physical contact, the enhancer and the promoter, okay? And all of this is to regulate the expression of the gene, okay? So turning the target gene on in a specific context in specific cell types, all right? And the other thing you need to know is that transcription factors recognize specific motifs in the DNA, okay? And we usually represent them by these logos, all right? So there's a sequence code, there's distinguished regulatory elements, and somehow, encoded in the genome is the program for how genes get turned on in specific cell types to maintain the identity of the cell and to respond to cues and do the work that the cell has to do. Okay, and then the other part is that this field that I'm in is driven by technology, and we have better and better assays to measure regulatory information. Okay, so one assay that's been really transformative are better ways of measuring regions of open chromatin. So where transcription factors are binding in the genome, these transcription factors are actually holding the chromatin open, and we can use assays to get sequencing assays to map those regions of open chromatin. One is called ATAC-seq, okay, so it's for assay for transposase accessible chromatin followed by sequencing, okay? And we've had that for many years where, these peaks that we can map correspond to these elements where transcription factors are binding, okay? But now we can do it in single cells, all right, single cell ATAC-seq, and that's also transformative, I think. Okay, the other part is that in recent years, this physical contact, this 3D interactions of chromatin, we are getting better and better at mapping them with assays like Hi-C, Hi-CAR, 4C, Hi-CHIP, okay, many 3D genomics assays. All right, so, and we can make these kind of contact maps of figuring out which genomic bins are interacting in 3D with other genomic bins, okay? And in particular, this helps us figure out which enhancers are physically contacting which promoters and figure out gene regulation. Okay, so the rapid improvements in these kind of single-cell epigenomics and 3D genomics technologies allow us new avenues for deploying machine learning to understand how gene regulation works. Okay, so with that picture in mind, I just want to motivate in a few slides why you should care about gene regulation. Okay, so where is all this being used, all these results, other than basic research? So I care about gene regulation because I care how cells work, and I try to understand the whole system. But even therapeutically, there is a reason to try to understand all of this and try to use machine learning. Okay, so you probably know we have cell-based therapies that are in the clinic, okay? And some examples are CAR-T therapy in cancer, right? So we take T cells out of the patient, we engineer them to express a chimeric antigen receptor that will target the cancer, lymphoma or myeloma. We infuse those engineered cells after activation back into the patient, and they kill the cancer cells, okay? Or the other one that's now in clinical trials, you start with pluripotent stem cells, right? What's being used is actually just a cell line, and you in vitro differentiate them, push them towards pancreatic progenitor cells that can further differentiate into beta cells that make insulin, right? You transplant these into patients, and it's a replacement therapy if the patient doesn't make enough insulin, right? So that's being used. Okay, so what is coming next? I think a lot of people are looking towards using understanding of gene regulation, understanding of enhancers, designing enhancers for control of transgenes. Okay, so this is a recent paper where, this is from Stein Artz's lab, and they are using a generative AI to design an enhancer that will control the expression of a transgene, both in fly and in human cells, right? And they're showing that they can make the gene express specifically in the brain, okay? They are presenting this just as a research tool, but therapeutically, people are thinking of, synthetic biology approaches where you will engineer the transgene to be expressed in a specific cell type, right, for safety and for precision. All right, so we're already thinking of designing enhancers. Okay. Okay, another motivation that a lot of people use is trying to functionalize, understand disease-associated genetic variation. Okay, so I don't know if you've seen these plots before. This is the summary of many genome-wide association studies, all right? So these are studies where you have a population with a disease, a control population, you genotype them, you do genome-wide statistical association between genetic variants, usually single nucleotide polymorphisms, and the trait, the disease, right? And you get a statistical association. Now, if you have a T instead of an A at this position, there's an association with a disease, right? And when I used to see these in talks, I thought this was a public relations exercise of the NIH after spending several $100 million on these studies. But it's actually quite impressive, right? We have almost 800,000 variants that are associated with traits. This is a lot of data. Okay, so the next part is, if all of these variants associated with disease changed the message, right? If they were encoding regions of the genome, we would know what to do. We would try to figure out how that altered message might alter function. But mostly they're not in genes, okay? Mostly they are non-coding, right? So, and there's a lot of correlation structure in genetic variation, but most of these risk alleles are not correlated with any coding variation. So what are they doing? Basically, the whole field thinks what they're doing is altering gene regulation. So these changes, these mostly point variants, single nucleotide polymorphisms, are changing enhancers, changing their activity, and changing the regulation of gene expression of specific targets and specific cell types, okay? So we have to understand gene regulation to understand how this genetic variation impacts disease, right? And if we can do that, then maybe that will give us some understanding and some target to go after, okay? Here's just a picture, if this is not all clear. I grabbed this yesterday while I was putting my slides together. Someone, here's an enhancer, here's a promoter. Maybe there's a little motif within the enhancer or promoter where a transcription factor likes to bind, and you change one nucleotide in this sequence or this promoter, and suddenly the transcription factors that like to bind don't bind as well or with high enough affinity, and instead of making transcript, you're turning off the gene or lowering it, right? It's not so simple, right? If it were just always ablating a motif, we would have figured more out by now. Often these variants are near motifs, but not in the motif, right? So there's sort of subtle variation, but it does something, okay? Okay, and one more motivation. So about 2 weeks ago, Alpha Genome came out. So this is Google DeepMind, big AI is now in this field. What they are doing is actually just a kind of scaled up version of models that have been out there. So this is Alpha Genome is a scaled up model version of Borzoi, a model from David Kelly's group at Calico. And what they do is they train a model. The input is a megabase of DNA. Okay, so you have your training examples are a megabase of DNA, and they train to predict thousands of epigenomic and transcriptomic tracks. Okay, so this is the kind of data we'll be talking about. ATAC-seq, so chromatin accessibility, but also gene expression measured by RNA-seq or CAGE, other kinds of epigenomic data or functional annotations, also contact maps from Hi-C. Okay, most of the prediction is at base pair resolution. Some of the assays are not at base pair resolution, and so they're 1/28 base pair or 2 kb. Okay, so you have this big model that is kind of producing multitask predictions of tracks, and what they do with it is they use it for variant effect prediction. So you, instead of the reference allele in the human genome, you put in the alternative and you get a change in prediction, and you interpret that change in prediction as sort of a functional, what does this SNP do? What does this variant do? Okay, and it seems like they're achieving state-of-the-art performance for variant effect prediction, which is still not perfect, but is, we're getting towards understanding what the non-coding genetic variants do. Okay, so that's kind of the background of what is happening in this field. Okay, what I'm going to talk about, I'm not going to talk about super heavyweight models of predicting everything. I'm going to talk about models where we take the data we have and we try to understand how genes are regulated, what enhancers are linked to what genes, how do they collaborate to regulate gene expression, and also some information about transregulation. Okay, so what transcription factors are affecting what genes in a dynamic process. Okay, so first I'm going to show you a model that uses single-cell attack to predict the contact map, and that gives you interactions between enhancers and promoters. Okay. Then I'm going to show a model called GraphReg or GraphReg Plus, the sort of new improved version, where we use graph attention networks to pass information over these 3D edges in the genome to predict gene expression. And then finally, this is unpublished work, but it's in bioRxiv, a model called Dynavello, which takes single-cell multiome data and learns a neural ODE so that we can get a functional form for an underlying vector field representing the dynamics of a system, and then we can intervene in the system, do in silico perturbations, and rescue experiments. Okay. So I think you know all the machine learning that I'm going to show you. I think it's the biology that's going to be hard, right? So I'm happy to answer any questions if things are not clear. And unfortunately, as it goes along, I think this is the most fun model, but it's also the biological context is the hardest. All right, so bear with me, but also please ask questions if anything about the problem or the biology or the motivation is unclear. Okay? Okay, let's start. So start with something relatively straightforward, predicting 3D genomic architecture and linking enhancers to genes. Okay, and we're going to do it by single-cell attack, predicting from single-cell attack alone. Okay. All right, so I need to give you a little bit of background on attack-seq and single-cell attack, just to give you some intuition for the data. If you've seen it before, you don't need this. If you haven't seen it, maybe this is all a lot, okay? But we've spent years looking at chromatin accessibility data, and this is a paper from a while ago, but it's still one of my favorite papers. It's with Andrea Schiedinger, and we were trying to understand why T cells that are specific to tumors just become dysfunctional and don't kill cancer cells, okay? And this was all done in a mouse model, and it's sort of the underlying epigenetic description of why T cells become exhausted or dysfunctional, okay? So, but for our purposes, it's just to give you, an intuition for looking at these tracks, okay? So we are looking at mouse T cells, And the functional T cells, naive effector memory cells, are here. And this is a liver tumor model, and we're watching as the T cells get dysfunctional, and they have the right TCR to kill cancer cells, but they don't kill cancer cells. The mice all get liver cancer. Okay? And we're looking at the locus that encodes PD-1 in the mouse. So this is immunotherapy target. And also interferon gamma. This is the effector cytokine that they need to kill. Okay? And what happens is you see that these accessibility tracks around the locus are changing. There are some events that are kind of on-off, okay? So like this enhancer that we and others described in a bunch of papers. This is an enhancer that's specific to exhaustion in the mouse, okay? But there are also other changes that are kind of more dynamic. You should have a big accessible enhancer and it becomes smaller. And then in interferon gamma, you have these other, something is missing, other things are changing. All of this is somehow encoding that we're turning on PD-1 really high, and we're turning off interferon gamma. Okay, so the accessibility changes at the locus are connected to gene expression changes that change what the T cells do. Okay, so just for some intuition of what this looks like. If you fast forward a few years, now we can do it all in single cells. Okay, so this is the same idea of let's look at T cell dysfunction, but now what we're doing is single cell attack, and this is looking at acute versus chronic infection. In chronic infection, you also have T cells that are dysfunctional in this different state. And we can look at specific loci. This is it's very sparse data, single cell data, but we can look at specific loci. And this is at the pseudobulk level, so we're taking a cluster of cells, looking at the locus, and piling up reads. And that same enhancer that we saw in bulk, that you only see in exhausted T cells, we can see it opening up in single cells. Okay? So that's just the intuition and some T cell stuff. Okay, so it's not enough for an enhancer to be open, okay? Accessibility doesn't tell you everything. It doesn't tell you about the activity of the enhancer. There are other marks that we would like to have to tell us more information. But also we need to know if it's connected in 3D with the promoter, okay? So we have other assays for that. These are 3D genomic assays like Hi-C. I don't know if anyone's seen this stuff before, but it's really cool. So basically, you're cross-linking the DNA in situ in the nucleus. You have a restriction enzyme digest, okay? You end up with, and then you ligate, glue together, and pull down. And what you get are these reads that are chimeric. Gray and blue mean they came from different regions of the genome, maybe far away in the 1D genome, but they were close enough to be ligated, right? So they were making a contact in 3D. And we call these reads contacts. And we map them to the genome. So the paired ends, we do paired end sequencing, and the pair of reads are mapped to bin I and bin J, and we say we have a contact between I and J, and we make a matrix, okay? So that looks like this, all right? And Hi-C data is fascinating, and people who think about structure and physics, have got into this field as well, okay? So what you see, if you take the contact matrix and just do a matrix rebalancing, you start seeing these, so you take the matrix and then you put it on the diagonal so you can line it up with genome tracks, okay? And you start seeing these triangles, okay, which are called topologically associating domains, and we think they're kind of like, these, the chromatin extrudes through a cohesin ring, and you have this self-interacting region, okay? But we actually process the data in a different way. We use a statistical background, negative binomial regression for a background model, and we can estimate the significance of these contacts. And then you can start drawing loops like this, like a graph. And we can see that, so this is actually the MEC locus. It's an oncogene. And it's very famous for having enhancers that are 2 megabases away. And you can see those enhancers and you can see those contacts. Okay, so we can connect enhancers in 3D with promoters. Okay, so Hi-C is cool, but to get this resolution, you need a lot of starting material. You need to do a lot of libraries. You need to sequence about 2 billion reads, and it's expensive, okay? So there's an idea that, single-cell attack, maybe there's already information about 3D connectivity in AttackSeek, okay? And the idea, there's sort of a method called Cicero from a while ago. And the reasoning is, maybe you see these peaks in single cells or metacells you have to kind of aggregate. And if they open and close together, if they're correlated in their accessibility, they're more likely to interact. And you can get a prediction of a 3D contact for free. Okay? And this is just an unsupervised, it's like a graph lasso approach on the contact matrix. Okay, so is it reasonable that we can just do a statistical procedure on the contact matrix? So what I'm showing you is the actual contact matrix of, this is about 2 megabases. Okay, you can see the sort of TADs. And this is the co-accessibility matrix from single-cell attack. Okay, and you can see, okay, there's some relationship between 3D organization and co-accessibility, but it's not perfect. This is another view. If you take true interactions versus all interactions, and you look at the distribution of co-accessibility, it's kind of shifted to the right. So there's a little signal, okay? So, all right, we have a lot of single-cell attack. We have some Hi-C. Maybe we can use a supervised model and just use deep learning. There's a subtle signal here. Maybe we can learn the relationship between single-cell attack and co-accessibility and Hi-C. Okay, so that's what we did. All right, so this is the model that came out last year. We are using this idea of co-accessibility and calculating it in the same way that the previous method did, okay? We're also sampling cells and generating a pseudobulk signal, okay? And we need a little bit of sequence information. So there are these structural proteins called CTCF that are associated with the boundaries of TADs. So we need a CTCF motif track, okay? And then it's a convolutional neural network, okay? So we are taking this information, we have a feature extractor, there's a kind of outer tensor, and then we also take this co-accessibility, we combine the two feature extractors, and we're predicting this V stripe. So along the genome, at 10 kb resolution, all the interactions forward and reverse in the genome up to 2 megbases. And then we glued the V stripes together, okay? All right, so again, we're trying to learn the contact map by a supervised model so that when we just have single-cell attack, we can make predictions for free, okay, about the contact map. And the contact map is otherwise pretty complicated to get, sometimes not feasible. So the first thing is a sanity check. Can we make predictions in held out chromosomes of the contact map? Okay, and that's what this looks like. Okay, So this is just a sanity check, right? A held out chromosome is not a real task. We want to be able to predict in new cell types. But here's the ground truth. Here's the prediction, okay? This is the motif track, the attack track, pseudobulk attack, and the coaccessibility, okay? And what you can see is it's actually pretty good, right? So this is a learnable signal. We can even predict through these there's these mappability artifacts in Hi-C, and we can make predictions there, okay? So, so this is good, but we don't want to just predict in held-out chromosomes. It's not a real problem to solve. What we want to be able to do is predict in new cell types or even new genomes, okay? So here we're taking the model that's just trained on human data, right? And we're making predictions with single-cell attack in mouse, so new genome and new cell types. This is an important gene, BCL6 in germinal center B cells, ground truth and prediction, and this is regulatory T cells, the Helios locus, another important gene, ground truth and prediction. Okay, and it's not perfect, but it's actually good enough that you can design experiments to test enhancers. Okay, so you can look at the promoter of the gene, see, so this is ground truth interactions with, and this is the prediction. It's good enough that you can actually take a look and test the enhancers that you get out of this prediction, and often you'll find an enhancer. So I think it's a useful tool for people trying to figure out how genes are regulated. Sometimes, to make good predictions, you need about 3,000 cells. All right, you can't do Hi-C with 3,000 cells, but if you have single-cell attack, and you identify different cell populations, you can make these predictions. Okay. One more application. Okay, so suppose you have bulk Hi-C and single-cell attack in a complex tissue like pancreatic islets where you have multiple cell types. So what you can do is fine-tune the model with the single-cell attack and Hi-C that you have, and then you can make predictions in individual cell types like alpha cells and beta cells, okay? And that's what we're doing here. So we fine-tune the model, and we have this ground truth islet bulk Hi-C, and then we make predictions in alpha cells and beta cells, okay? And for a long time, we didn't know how to validate these predictions, but someone actually did the experiment, sorting out alpha cells and beta cells, and did the Hi-C, and then we could compare. And it's not perfect, okay? But these are alpha cell ground truth and prediction, beta cell ground truth and prediction. And what I'm showing is that you can see these cell type specific enhancers of glucagon in the alpha cells and of insulin in the beta cells. It probably can't see it very well. Okay, so again, it's not perfect, but it's good enough to work with and give you some information, predictions of what enhancers are talking to your promoter. Okay, so that's the first thing. This is very concrete. So the next part is trying to understand gene regulation in a system in pancreatic differentiation, and using 1D and 3D information. Okay, and again, I'm going to show you a bunch of different models in the context of this project. It's sort of written more about the, trying to get information about the system, but I think you'll see there's a lot of machine learning that we are throwing at the system. Okay, so I actually mentioned before this protocol that you can take pluripotent stem cells and in a kind of multi-step procedure, differentiate them in vitro into beta cells that secrete insulin. Okay, this is Doug Melton's procedure. And this is also, this protocol is now being used by Vertex Pharmaceuticals. It's in clinical trials for type 1 diabetes. Okay, so what we have is these different stages of this in vitro pancreatic differentiation. We have a 3D genomic assay called HiCAR, okay, a little more efficient than Hi-C. We also have single-cell multiome data at these different stages. So single-cell multiome, we have chromatin accessibility and RNA from the same individual cells, so 2 readouts from each cell. And again, at these different stages of differentiation. So the question is, can we link enhancers to genes? Can we interpret non-coding genetic variants that are associated with type 2 diabetes? Okay, so some of the pathology in type 2 diabetes is associated with deficiency in the pancreas, right, in the endocrine cells of the pancreas. Not all. And then can we build predictive models of gene regulation from the 3D genomics and the 1D information that we have? Okay. Okay, and here's an example. Again, lots of genes and biology, but so what we're seeing is different stages of this differentiation process. This is human ES cells, definitive endoderm, pancreatic progenitor 1. And what we're seeing is these enhancers of a particular gene, which is it's a transcription factor, PDX1. It's a developmentally important transcription factor. It's a diabetes gene, okay? And it's gaining 3D interaction with its enhancers through differentiation. So you're seeing enhancer rewiring and differentiation. Okay, so this is kind of the data we have. Okay, so I'm going to show you a bunch of different models. I think we are overwhelming our collaborators with these different approaches. So now we have a lot of tools, and we're trying to understand this very cool data set. All right, so I'm going to just show you a bit of the multiome data. Okay, so again, we have, so this is the protocol. So it's a 7-stage protocol. It takes a long time. You start with pluripotent stem cells, definitive endoderm. We're skipping the foregut or gut tube, pancreatic progenitor 1, 2, and then like these early and late endocrine stages. Okay, and I'm showing you a UMAP of the RNA and also of the attack. Okay, so we have these 2 readouts from single cells, but we're looking at them separately. And in particular, it's not a very fine-grained time course, right? The early stages, the cell types are very different from each other. But what's cool is starting at PP2, there's a population called NeurG3 positive cells that separate off from the rest, and they kind of seem to give rise to the endocrine cells. Okay, we're not using a standard embedding. We're using an embedding that we developed called cell space, and I'll just show you one slide on how that works. Okay. All right, so the idea is we wanted a sequence-informed embedding of single-cell attack, okay? And the reason is this data is very sparse, okay? You can think you have several 100,000 accessible peaks, right, represented in your cells. It's much higher dimensional and sparser than single-cell RNA. The dynamic range is, for any event is 0, 1, or 2, because you only have 2 chances of seeing an accessible event in a diploid genome. Okay, and there are actually complex batch effects. So we thought that if we use the actual sequence information that's in accessible sites, we could mitigate batch effects and get a better, more intrinsic representation, okay? Because the sequence information contains information about motifs that transcription factors recognize. Okay, so what we do is we learn an embedding where cells and DNA chimeras are embedded to the same latent space and inform each other's embedding, okay? So briefly, we sample sequences from accessible events. We turn that sample into a bag of chimeras, okay? Not quite, but almost a bag of chimeras. And then we embed the chimeras and push them towards the cells in which they are open and away from the cells in which that region is closed, okay? So, and then it's just it's a margin-based ranking loss. Okay, so we're pushing cells and chimeras around. In the end, all DNA chimeras are embedded and all cells are embedded. And the algorithm is actually star space. It's from Facebook AI. It was originally for embedding tweets and hashtags, right? So yeah, but we saw that it could be used for this. Okay, so just showing you can learn this structure and you can kind of see this progression to, early beta cells, late beta cells, or early enterochromaffin, late EC. Okay, so I think actually there's a lot of cool information in the single cell attack. One more algorithm, it's very simple, okay? So this also came out last year. If you have 2 readouts, right? If you have a single cell attack and single cell RNA from the same cells, every cell is a training example, and you can learn, this is just a, 1 gene at a time model, you can learn to predict gene expression from chromatin accessibility, okay? So that's what this is supposed to show. We're just learning a Poisson regression model, okay, very simple. We're taking the gene that we're trying to model, we look at just tiles, genomic tiles across the gene body and flanking regions, and we're learning this regression model that predicts gene expression, so predicting gene expression in single cells from chromatin accessibility in single cells. And then we can use just Shapley values in single cells, aggregate over cell types, and identify cell type specific enhancers, okay? So doing feature attribution to figure out from this general model, what are the enhancers that are active, that might be active in individual cell types, and then see if we can annotate fine-mapped disease-associated variants. Okay, everyone with me? So here's just an example of that, okay? All right, so the regression model's at the bottom. We're modeling a particular gene called GLIS3, another diabetes gene. The gene is in red, okay, going this way. And then we have these flanking regions. And what I'm showing is, so this is a single cell model, but I'm showing the pseudobulk attack over different cell types and also the distribution of expression over those different cell types, okay? And I don't know if you can see it, but these dots with red circles come from Shapley value analysis. These are enhancers that are specific to cell types, so we can figure out from just this simple model which enhancers are important for predicting gene expression in specific cell types. Okay, and in particular, there's this region with 3 diabetes-associated SNPs, and we can see it's important in specific cell types. It's in the first intron of the gene. It's about 10 kb from the transcription start site. So we can home in on what cell types maybe these genetic variants are affecting. Okay. Okay, so what I really want to talk about in this section is using the 3D information together with chromatin accessibility to model gene expression. Okay, and we're going to do this at the pseudobulk level, meaning we have our single cell multiome data, and we're going to aggregate over cell types. Okay, it's not a single cell model yet. And we went back to a model called GraphReg that we published a few years ago. And I'm very fond of this model. I think it's kind of a nice, midway point between models that predict everything, like AlphaGenome, and models that are very, local. This is a large-scale model in terms of genomic context, but we're training just one cell type at a time. Okay, so the idea is we think of the genome as a sequence of bins with edges between them, right, that come from Hi-C. So the edges are 3D contacts, right? So we think of the genome as a graph, and then we want to pass information along those edges between enhancers and promoters, and supervise to predict gene expression. Okay, in this paper, there are 2 versions of the model. Okay, one model just started with epigenomic data. It's over a 6 megabase region, so you have these tracks, accessibility, and a few histone marks. And what we're trying to predict is a readout of gene expression called CAGE. Okay, CAGE tells you it's a 5' capture, so you're kind of mapping the transcription start sites, and it's a kind of you're getting a peak wherever a transcription start site is being used. So it's kind of a more quantitative and easier gene expression readout to deal with. So you have these kind of peaks. So what we do is we start with a regular convolutional neural network. So we're taking the 1D epigenomic data and learning representations of it. Okay, so kind of compressing the information. Then we pass this representation to a graph attention network where we're passing information along these 3D edges, the 3D contact edges. And ultimately, we predict at 5 kb resolution these CAGE peaks, the activity of transcription start sites. Okay, so that's the first model. And then you can imagine we can take this model and we can do feature attribution for specific genes, figure out what enhancers are important. Okay. So there's another version of this model that starts with DNA sequence. It's actually the biggest sequence model there is still. We're starting with one hot encoding of 6 megabases of DNA. The first part through the model is just a dilated CNN. Okay, so we're doing regular CNN and then a kind of dilated convolutional layers and predicting the epigenomic tracks. Okay, so you can think of this as trying to learn motif-like representations that help you predict the 1D epigenomic data. Okay, and then this motif-like representations we're feeding to the graph attention network, passing information between enhancers and promoters, and then predicting this gene expression that's, this transcription start site activity called CAGE. Okay? All right. So one figure I'd just like to show the difference between the graph attention network and dilated convolutional neural networks. Okay, so this is back to the MYC gene. You remember, so MYC is this gene. It has enhancers that are 2 megabases away, right? And what I'm showing in yellow are predictions and then the feature attribution for the graph attention models, okay, if you use this 3D information. And in blue, the prediction and feature attribution for just dilated CNNs, which were the state of the art at the time. And what you can see is that only the graph attention network can correctly learn that there are these very distal enhancers that are important for the expression of MYC, right? And you can see why they can learn it, because we can reach very far away in the genome over these edges and learn this information is important. Whereas dilated CNNs, even though in principle they have a big enough receptive field to see the whole sequence context, in fact, all the information decays away and you only see information near the promoters. This is actually true of the transformer models as well, that they underweight information from enhancers and focus on promoter, like proximal region. And I think it's true even of the fanciest models, okay? So, this graph information from Hi-C is actually very useful, right? And once you see it, right, you see that it's useful, right? So, okay. So what do we need to do now? Okay, so we're back to pancreatic differentiation. We're trying to understand type 2 diabetes and the genetic variants that have been associated with type 2 diabetes. We don't have all those histone marks that, that we used previously. All we have is the pseudobulk chromatin accessibility, pseudobulk attack, and we have RNA, which is a 3' capture, which doesn't tell you about transcription start sites. It's the other end, right? So we only have expression at the gene level. So we need to make some changes. So the first thing is we just extend the graph. So for every gene, we look at all the annotated transcription start sites it has, and connect the gene to those genomic bins. Okay, so we're kind of going to supervise at the gene level instead of the bin level, and we have to kind of have an extended graph and also use graph attention on this part. The other thing is the attack-seq is not a very rich representation. It's not as rich as having all these different histone marks as well. So what we do is sort of 2-part. We first learn a model that goes from DNA sequence to chromatin accessibility, DNA to attack. We take the representation from that model and concatenate it with the attack-seq and use that as the representation. Okay, so so representation from a sequence model plus attack-seq signal, and that's our input, and that works well. Okay, and now we can do this model and train pseudobulk and start looking at feature attribution. So this is just gradient by input feature attribution. So sorry, it's very messy, right? So this is every cell type. I'm showing the 3D interactions with all these complicated loops, and the red is the feature attribution. And so, for example, here we're doing feature attribution for a gene called 1cut1, and we can find in these cell types importance of an enhancer that contains a type 2 diabetes risk SNP, and it's more than 600 kb away from this transcription start site, but we can see its connectivity, and the model can figure out it's important, okay? And in fact, this is validated. There's another paper that explores what this SNP is doing, and we can figure out what transcription factor it is that's affected. So this is kind of just an example where we know that we got the right answer. Here's another example where that's the PDX1 example that I showed before, and this is another enhancer containing a T2D risk SNP. It's only 40 kb or so from the transcription start site, but we can see that it's important at all the later stages. Okay, so it's a modeling system that allows you to actually see enhancers connected to genes important for regulating genes that are pretty far away. And we can look at fine mapped variants and try to associate with genes and with cell types. Okay. One more slide on this. So just for a bit more quantitative, how well is this working? We need a bunch of validated enhancers and non-enhancers to assess. And there's a nice screen from our, all this work was done in collaboration with Danwei Huangfu at Sloan Kettering, and she has these beautiful screens she can do. So the idea is that you do differentiation, okay? So you do differentiation from pluripotent stem cells to DE. And then you do a CRISPR-I screen against candidate enhancers for important developmental genes. And if you succeed in turning off an important enhancer, it will cause a defect in differentiation, and that's what you can screen for, okay? So this is testing 394 enhancers, and there were 24 positives associated with this defect in differentiation for 4 different genes, okay? So we know the gene, we know the enhancer, and we can see if GraphRay correctly models it. Okay, and this is kind of this score out of the model from feature attribution for the true positives versus the true negatives, and basically the majority of these true positives are detected, even at pretty large genomic distance. There are some that are missed, and sometimes they're missed because the high C is not telling us that they're connected, and that could be a sort of capture issue in the high C, okay? But it's actually not too bad. I think at the AUROC is like 0.77, it's not so bad. Okay. I want to pause here before I go into the last section and see, are there any questions so far? Because I know, yes.
Now? Okay.
That works.
Yeah, so while for the first model, it was quite obvious to me that we want to replace long, potentially expensive assay with the model predictions. Could you summarize how many time and cost savings GraphRec gives us?
So, it's true. There's sort of these 2 different motivations in this field, right? So sometimes you have a model where you're making a prediction to save you doing an experiment, right? So and maybe it saves you doing an experiment that isn't even feasible, okay? So if you have a rare cell population, you wouldn't even be able to collect the cells. It would be very painful to collect the cells and try to do the high-C, right? So that's chromofold, right? So very straightforward. For the GraphReg model, for this stuff, and for alpha genome, you are using all the data you have, right, and interpreting the data, right? So it's a little different, right? And the only, so if you're using DNA sequence as an input, you're learning information that's specific to that cell type, and it doesn't easily generalize to new cell types. And that's true of alpha genome and Borzoi and Informer and all the models that came before, right? The only novel prediction that you're making is I can change the sequence, right? So I can say, here's my reference sequence. They're all trained on the reference genome, and I can change the reference variant for the alternative and see if my prediction changes. So that's the variant effect. Or I can, make more severe edits, or I can do in silico mutagenesis, feature attribution, figure out where the information is. So these are interpretive models, right? Figuring out what information is important, right? And then it guides the next experiment. So instead of doing an enhancer CRISPR-I screen for every gene that you care about, right? And every cell type you care about, you can use the model and home in on specific candidate enhancers. So that, you interpret the information and you can use it as a sequence to gene expression model. What happens if I change the sequence, scoring variants, okay? We haven't really, I'm not sure that GraphReg is the best variant effect predictor, just because this sequence model is pretty standard. We haven't really assessed, but I think it's pretty good at finding distal enhancers that really regulate genes. Yeah. So yeah, it's true. We're not always just making predictions to save money. It's saving money in that we understand our data and we have a better idea of what the next experiment should be, right? But we're using all the data we have. Yeah. Yeah. Other questions? Okay, so no other questions. I'm going to do one more thing. I think I still have time. Okay, so learning cellular dynamics from single-cell multiome data. So this is the newest work. I'm excited about it. It's a lot, maybe, biologically, but let me give it a try. Okay, so if you know about the single-cell world a little bit, you know that the measurements are destructive, right? So this is single-cell transcriptomic data, single-cell RNA data from a bone marrow, right? Human bone marrow donor. So we've taken the cells out of the system, dissociate them, put them through this kind of microfluidic device and collect single-cell RNA, and now the cells are gone, right? And really, we don't have these high-plexity measurements in vivo, okay? And at best, we can do imaging in a dish, right? So for now, this is what we have. Very good data, but we've destroyed the cells. And often we're trying to understand some underlying dynamic process, like a developmental process. Like in the bone marrow, you have hematopoietic progenitor cells that give rise to all the adult blood cells, and this happens every day, right? But we don't have time series data, and we don't have, people are trying to do lineage tracing. It's very complicated. So what we have usually is just a single snapshot where, one time point, okay, we have a single time point where we think some cells are earlier in a process and some cells are later in a process. And we're trying to learn a pseudo-temporal ordering, pseudo-time, or trajectories. And there are a zillion of these methods, okay? So this is one I like, actually, and this is from Donna Pear's lab, but Manu was my former student, so Manu Sethi, and, so you need to specify a starting point, and it does a Markov process on this underlying graph and kind of infers this pseudo-temporal ordering, and you can get some trajectories out of this. Okay. All right, we want to do a bit better, and we are motivated by RNA velocity. I don't know if anyone's heard of this. Okay, but we're not going to exactly use the details of RNA velocity. It's just the idea of writing down a differential equation. Okay. So what RNA velocity does is, again, single snapshot, but and it's for single cell RNA specifically. And it says, well, maybe there's something about time's arrow, okay, in just looking at, in order to turn on a gene, first you turn it on and you'll see some unspliced reads because you'll have a transcript which hasn't been fully spliced and it's not mature. And then later on, you'll have more mature transcripts. Okay, so they're taking these single cell measurements very hard. Okay, some of the reads you can say they're unspliced, and some are from exons and they're spliced. They write down a differential equation. They say you should have a phase diagram like this. You turn on the genes, you see unspliced first, and then more spliced, and then you turn off the gene, you reverse. Okay. And then from this ODE, you can get a rate of change in single cells of the mature transcript. And if you can do that for enough genes, you can get a vector field, okay? All right, many caveats. First, if you actually look at these phase diagrams, it never looks like this for real data. It's kind of just like a blob, right? So it's all, and this isn't even right biologically, right? So, but it's cool, right? It's cool. The idea of estimating a vector field is cool. There's sort of issues of robustness. You can only compute this for a subset of genes. You're only doing this ODE one gene at a time, and then you have to do a bunch of steps to get a latent time for all the genes. So there's a whole pipeline. Okay, but the idea that there's an underlying ODE governing the system, there's an underlying vector field that you could learn from the data, that's the idea we want. Okay, so, and you'll see how, at the end, how it's connected to drug discovery. Okay. All right, I know we're going pretty far, but all right, we're going to do it for multiome data because we think that the attack data is actually giving you additional information in some systems, okay? But as I mentioned, chromatin accessibility data is very high dimensional, and we want to compress the information in a way that will still be interpretable. And we do that by getting motif accessibility. Okay, so for every cell, we can get a library of motifs, and for each motif, we can kind of, relative to a peak atlas, figure out the accessibility of the motif and give a score, okay? Looks like this, CBP alpha has this motif. We scored against, in single cells against an atlas of peaks. There are certain hits, and there are some cells where that motif is very accessible, and there are other cells where the motif isn't accessible, okay? So motif accessibility, that's the first, those are the features for attack. The features for RNA are just the gene expression. And the idea is that transcription factor motif accessibility and genes regulate each other in a dynamic process. And we're going to learn a functional form of the RNA and motif velocity vector fields and the latent times for all cells directly from the data using a neural ODE. Okay, so this is the idea, okay? So every cell is represented by gene expression and a bunch of motif accessibility. We're using an encoder-decoder formulation. It's actually a generative model, okay? So we encode into a latent space, and we learn the dynamics with the neural ODE in the latent space, and then we decode. Okay, so what are we actually learning? We're learning the starting point in the latent space, the RNA latent space and attack latent space for every cell, and then the ODE, like the integral curve, tells us where the cell ends up at time t, and this time t is the latent time that the model is assigning to every cell. Okay, oh, I forgot to add the loss function. So the loss function, there's many aspects to the loss function, right? So there's reconstruction loss, there's, we want the vector field and in the RNA space and the motif space to be consistent. And then we also, we supervise with RNA velocity. Okay, so I mentioned RNA velocity is not perfect. Sometimes it doesn't work at all. But for some genes, we have an estimate of its rate of change in every cell. And so we can just try to make our velocities consistent with RNA velocity. Okay. So this is the model. I think I've said everything. Decode back. Yes. Okay. And there's a preprint. Okay. Okay, so this is the model. And then we thought, there have been so many of these papers, except not in multiome, okay? So luckily not in multiome. We need to do something really hard, okay? So what we decided to do is model the dynamics of B cells going through the germinal center reaction. Okay, so germinal centers are these transient structures in your secondary lymphoid organs, right? And it's part of how B cells modify their B cell receptors and differentiate to plasma cells that secrete the antibodies that fight infection, okay? So it's really important, right? And what happens, it's complicated, right? So in the germinal centers, there are these dark zones where the cells proliferate and undergo somatic hypermutation of their antigen receptors. So they're editing their own genome, crazy, right? And then they migrate to the light zone and interact with other cells, T follicular helper cells and follicular dendritic cells. And if it looks like the antigen receptor is good, they get T cell help and survive, otherwise they die. But then they might go back, for another round. So there's sort of this cycling, and then they can exit either as kind of plasmablasts, and they'll eventually become plasma cells that just are antibody factories, or they become pre-memory B cells, so the memory B cells are for the next infection that, they're ready. Okay, so complicated dynamics, cycling, multiple exits, and transcription factors, we know, govern all the steps. Okay, so maybe the attack readout is important here, and this motif accessibility is important. Okay, so yes, what can we do? So quickly, this is a visualization in RNA and motif space of the dynamics that we learn, okay? So green is in the dark zone, so you can see this basic bifurcation of stay in the dark zone and proliferate or migrate to the light zone, the blue cells, and then you can see exit as pre-memory B cells or plasmablasts, okay? So you can even see this recycling in orange, like you were here and you went back into the dark zone, okay? So this basic bifurcation we're learning, which looks good. This is the latent time, so start here and go here and here. I think SC Velo is here learning the wrong thing, okay? Because it looks like everything is exiting, right? So it's not learning this bifurcation. Okay, again, quickly, since I know we're running out of time. Dynavelo, like other methods, you can order cells by pseudotime or latent time and start seeing patterns. I'm showing patterns only for transcription factors, and the main thing I want you to notice, because the slide is too complicated, is that you can see more dynamics in the transcription factor accessibility than you can see at the RNA level. Okay, so transcription factors aren't always regulated at the RNA level. It's kind of, are they active? Are they going to their sites? Are they in the nucleus? And you can kind of see, the pink-green is the motif accessibility, and the blue is gene expression. And sometimes the blue is very subtle changes. But we are finding the right kind of transcription factors along the right trajectories. Very quickly, okay, we also have mutants. These are all mouse, okay, mouse germinal center B cells from Ari Melnick's lab. We have mutant cells. ARID1A heterozygous loss is an event that you see in B-cell lymphomas, okay? So we can do the model in mutants, and we can start seeing how the trajectories change when you lose the activity of a transcription factor. So for example, now it looks like IRF8 is more important than in wild type. We also have the CTCF heterozygous loss, okay? So certain things change, and we can model that, and that's kind of interesting. A bit more interesting, we have a functional form for the dynamics, which means we can do Jacobian analysis, right? So we can ask how, for example, the expression change of one gene changes the RNA velocities of other genes. Or we can do an in silico perturbation, in silico knockdown. So if we have a transcription factor, we can set it to its minimum expression value and its minimum motif accessibility across all genes and see how all the other velocities change, okay? And that gives us edges in a regulatory network, like this transcription factor affects this gene, okay? And those edges are dynamic along pseudotime. So, these transcription factors, their targets are only, in certain cell types, and then they're not important in later cell types, and we can model that. Okay, so that's what I'm showing. Regulatory edges from Jacobian analysis or in silico perturbation, but showing it along pseudotime and in wild type and mutants, okay? And from that, we can actually extract about everything that is known about the germinal center in terms of transcription factors and their targets just from this analysis. And we can also make predictions about what edges are lost in these specific mutants, one of which is cancer relevant, okay? So it gives us some idea of where to intervene in the system. 2 more slides, sorry. Okay, so I showed you wild type, I showed you this heterozygous loss. Can you actually take the model and predict what changes under a genetic loss of function, okay, from the wild type? So suppose you have the wild type model, okay, and you do in silico perturbation for ARID1A, right? You do an ARID1A knockout, and you're trying to see what changes. And does it actually look like the ARID1A, loss? Okay, and sort of, okay? Like you actually do get some effects. So if you look at the delta velocities, okay, you can kind of see some changes, like maybe we're, exiting more and we're losing the recycling, and that's kind of phenotypically what happens in this loss. Everything exits the germinal center reaction as immature pre-memory B cells. But we can also do a statistical test and ask, in different cell types, when, what velocities, RNA velocities and motif velocities are significantly changing under this in silico perturbation compared to the ground truth, and see that some of the things are actually right, like this more important RF8 is, more important in this knockdown of ARID1A. Okay, last slide. If you have this defect, can you predict the perturbation that will rescue and turn the dynamics back to more wild type setting? Okay, so here we're doing the same thing. We test transcription factor in silico knockdowns, and we ask, how many RNA velocities and motif velocities would it rescue? Okay, would we predict to rescue? Just doing a statistical test again. And so, for example, we predict that a tox knockdown would be predicted to kind of rescue the dynamics of ARID1A loss. And our collaborators think that this is reasonable, but it's not validated fully yet. There's another prediction that is validated, but I'm not allowed to talk about it because my collaborators are nervous and this is being recorded. But that's what I wanted to tell you, okay? So you can see that this kind of modeling does enable understanding a system, perhaps, so that you could intervene and get a target, though the targets are transcription factors. Okay, so if there's any time, maybe there's not, I'm happy to take more questions, and I just want to acknowledge all the amazing postdocs and grad students who did all this work.
Thank you so much for the great presentation. I was curious, so I guess one underlying assumption of this RNA velocity type modeling is that the RNA actually specifies the entirety of the dynamic state of the cell. To what degree do you think that is true, and could you give a number to that? Is it 10% correlated with the entire state of the cell? Is it 90%? Where do you think it is?
I mean, I think that we wouldn't be able to learn all of this with just the RNA velocity. I think the motif velocities are important, and I think that we don't have all the measurements that we really need, right? But we have what's easy to collect. So I think in this system in particular, I don't have a a prediction or a, assessment of what the RNA captures. I'll just say, the RNA, expression changes can be caused by many things, and things are, conflated, right? And also, there's certain transcripts that you don't capture very well. Like, there's also all sorts of technical reasons why the RNA is incomplete and noisy as well.
Yeah, that was going to be my follow-up question then, because even if you believe the RNA sequencing covers a lot of the dynamics, to what degree do the measurements that we have actually cover the RNA? Because we know from the scRNA-seq, for example, mostly low read depth, so low transcript expression, probably not well reflected, and we know some low expressed transcripts have outsized effects. To what degree actually do you think we're able to really truly capture the dynamics yet? And do you have like a wish list in terms of what new technologies could help us do this?
I mean, I actually think the ATAC-seq is more important for capturing differentiation states of cells. So I think that, it's already, I have my wish list there. It would be nice to have nuclear kind of protein level measurements of transcription factors, that kind of thing. It's not there are some technologies, right? But we learn a lot from RNA, right? The single cell, I don't want to sort of say the single cell RNA is, it's sparse, it's noisy, but we know how to analyze it, and it's empowered a lot of research, including discovery of cell types in the immune system that we didn't know about 3 years ago, right? So it's important, right? Yeah.
For sure, thank you. It's exciting. Thank you.
Any other questions? So maybe I have one.
Okay.
Because you mentioned that you have two modalities of data in the second model, and
Yes.
You are trying to predict one of the modalities from the other. And have you tried any contrastive learning, like vision language models, to see whether this approach can be used there too? Or there is, this doesn't make sense from the motivation perspective.
We haven't done more than we've done. I mean, I think what we're saying is just this, that the rate of change of RNA depends not only on the current expression state of the cell, but also the current chromatin accessibility. And similarly, the rate of change of the motif accessibility depends on both, so that they're influencing each other. But beyond that, we're agnostic. And the other thing is we're, not modeling the details of gene regulation. It's kind of a global trans effect of the transcription factors. We're not encoding which transcription factors could plausibly regulate individual genes. That would be combining everything I showed you into one model, which we haven't done. So.
Okay,
Yeah.
thank you. So
Yeah.
let's thank Christina once more, and now we will have a short break.